In this video, we will describe the activity of topoisomerases, specifically focusing on class 1 topoisomerases. Remember that class 1 topoisomerases alter the linking number by cleaving and religating only one strand of DNA. Topoisomerase 1 can be further categorized into two groups based on differences in their mechanism of action, topoisomerase 1A and topoisomerase 1B. Let's examine the differences and similarities between these two topoisomerases. If we look at the active form, both topoisomerase 1A and 1B are monomeric, which means that they consist of only one subunit. We've observed that both enzymes cleave and reseal one strand of DNA during their activity. This process involves the formation of a transient covalent bond between a catalytic tyrosine in the enzyme active site and a phosphate group at one end of the cleaved DNA strand. It's known as a phosphotyrosine bond. Because only one strand is cleaved, only one phosphotyrosine bond will form. Notably, this process does not require ATP. The action of the two topoisomerases differs slightly. Topoisomerase 1A is capable of removing negative supercoils from DNA, while topoisomerase 1B can remove both positive and negative supercoils. One major difference lies in their mode of activity or mechanism of action. Topoisomerase 1A operates through a mechanism known as strand passage, while topoisomerase 1B follows the controlled rotation model. Let's examine how topoisomerases 1A and 1B cleave a single strand of DNA. In the first step, the topoisomerase 1 enzyme binds to the DNA. The hydroxyl group of the catalytic tyrosine residue initiates an attack of a phosphodiester linkage in one of the DNA strands. The result is the cleavage of this DNA strand. A transient phosphotyrosine bond is formed, and once this strand is cleaved, the other strand can pass through the gap that the broken strand created, which alters the twist number. Finally, at the end of the process, the phosphodiester bond is cleaved. Simultaneously, the phosphodiester linkage is restored in the broken strand. At this point, the topoisomerase action is complete. Let's examine the structure of the topoisomerase 1A enzyme. Here we have a partial structure that displays domains 1, 2, 3, and 4, which collectively form the catalytic region of the topoisomerase. This region exhibits a shape resembling a C and is hence termed a toroidal structure. Domain 3 and domain 1 are on opposite ends of the C structure and can move relative to one another. Their movements contribute to reversible conformational changes from an open to a closed structure. In the closed structure, domains 3 and 1 are in close contact. Furthermore, domain 4 contains a groove that displays basic residues. This positively charged groove can engage in electrostatic interactions with the negatively charged DNA strands. Finally, the cavity that's formed by these four domains in the closed conformation is spacious enough to accommodate a single strand of DNA, but too small to host double-stranded DNA. Now let's focus on the strand passage model. In the initial step, topoisomerase 1A binds to one strand of DNA in an underwound region of the double helix where the two strands are separated. A conformational change from an open to a closed structure drives this process. As this region closes, bringing domain 3 and domain 1 closer to each other, the enzyme pinches one of the DNA strands, which positions a catalytic tyrosine from domain 3 near the DNA strand. This catalytic tyrosine triggers the formation of a phosphotyrosine bond between the topoisomerase's domain 3 and the 5' end of the cleaved strand. The subsequent step involves a change in the structure's conformation from closed to open. Domain 3 moves away from domain 1 and creates a gap between the two ends of the cleaved strand. The cleaved end of the DNA strand covalently bonds to domain 3 and moves away from the other part of the cleaved strand, forming electrostatic interactions with the positively charged groove on domain 4. The intact strand that hasn't interacted with topoisomerase 1A yet moves through the gap and enters the cavity. The topoisomerase 1A undergoes another conformational change, transitioning from an open to a closed conformation, which brings the two ends of the cleaved strand back together. At this point, the phosphotyrosine bond between domain 3 and the cleaved DNA strand is broken, and a new phosphodiester bond is restored. This effectively repairs the cleaved strand. The final step involves another conformational change, returning to the open conformation. Ultimately, this process alters the twist number by 1. Subsequently, the enzyme dissociates, which releases the DNA and prepares to initiate a new catalytic cycle. In contrast to topoisomerase 1A, the mechanism of action of topoisomerase 1B involves a different structural arrangement. The structure of topoisomerase 1B's catalytic domain differs notably. It comprises 3 subdomains, domain 1, 2, and 3, along with a C-terminal domain. In the side view, it may not be immediately apparent how the structure interacts with DNA. However, when viewed from the front, domains 3 and 2 are observed to be nearby and form a cavity within the catalytic domain of topoisomerase 1B. This cavity measures approximately 20 angstroms in diameter, which suggests its capability to bind double-stranded DNA. When considering the structure of this enzyme bound to DNA, it's clear that the double helix of DNA fits snugly inside the structural domain of topoisomerase 1B. This alignment suggests a potential mechanism for interaction between the enzyme and the DNA molecule. Let's delve into the mechanism of action. Initially, subdomains 2 and 3 are far enough apart to allow the double-stranded DNA molecule to enter the central cavity. Upon binding, we observe a non-covalent complex initially formed between the DNA and the enzyme. The C-terminal subdomain contains the catalytic tyrosine, which triggers the cleavage of the phosphodiester linkage on the top strand of the DNA. This action results in the formation of a phosphotyrosine bond. Notably, instead of the phosphate remaining attached to the 5' end of the strand, it remains associated with the 3' end of the strand. Consequently, one end of the strand becomes permanently attached to the enzyme, while the other end is free to rotate about the intact bottom strand. Multiple controlled rotations occur within one catalytic cycle. The rotation of the strand around the other strand is facilitated by the close contact between the DNA and the cavity of topoisomerase 1B. The strand can attach to one point inside the cavity, detach, and then attach to another point, thus facilitating controlled rotation. After a certain number of rotations within the catalytic cycle, rotation ceases, and the phosphotyrosine linkage is cleaved. Subsequently, the phosphodiester linkage is reformed, which results in the restoration of the non-covalent complex. Most topoisomerase 1 are ATP-independent enzymes that relax positively and negatively supercoiled DNA. To conclude, the topoisomerization of DNA molecules can be catalyzed by class 1 topoisomerases, which include both topoisomerase 1A and topoisomerase 1B. These enzymes cleave a single strand of DNA, forming a transient phosphotyrosine bond. This action leads to a change in the twist number, consequently altering the linkage number of the DNA molecule. This alteration can result in the removal of either negative or positive supercoils. However, it remains unknown what the precise mechanism that governs how many times the topoisomerase 1 enzymes act on a DNA molecule is, and thus how many positive or negative supercoils are removed. The exact regulation of this process is not well determined.